All right. Well, great. Max comes to us as the greatest world traveler for today's event. Max is. Oh wait, you guys filled out little introduction cards. What does Max's card say? Max Lindell is a research scientist at the Medical University of Vienna. Started using Dragonfly lightly in 2020 and then routinely in 2021. Max is part of a collaboration I work with researchers spanning a few different institutes. They look at a really interesting biological system. And Max is going to tell us about his experience using deep learning. He's going to talk about segmentation and analysis of cellular ultrastructure of retinal pigment epithelium. So let's look at these cells on the back of your retina.
Okay, hi everyone, I'm Max, and I do come from Austria. I'm an American though, so that's why I don't have an accent. And I'll be telling you about our work reconstructing an individual cell from the retina. And for anyone who doesn't know, the retina is a thin layer of cells inside of your eye, and light passes through the lens and hits the back of the eye, and that's where the magic happens. So that's how you're all seeing me. If someone doesn't have a retina, please see me afterwards, and I'd be very interested to know how you're doing this. But probably everyone has a retina, and our research is focused on this thin layer of cells right outside of the rods and cones, which a lot of you probably have heard before. And this RPE, or retinal pigment epithelial cell layer, is it supports these cells, the rods and cones, and it nourishes them and maintains their health. So if we look a bit closer at these cells, they have processes or microvilli that extend into the space around the photoreceptors, and they wrap around and attach to them. And basically what happens in the visual cycle is that, without going too much into depth, you produce a bit of waste. So that needs to be recycled, and that's what these RPE cells do, is that they grab the shed discs of the photoreceptors and all the other waste and recycle it through their body and then pump it back up into the system. So this all is very important for maintaining the health and a proper visual function. And this can be considered the start of the visual process. This is where the proverbial magic happens, and it is part of the central nervous system. It's highly compartmentalized, it's highly polarized, so it's not that clear yet. It will become later that there are certain areas in the cell that are always filled with more or less the same organelles, with the same components of the cell. And if we look a bit closer at these apical processes, or these microvilli, you can kind of see in this picture how they form around the photoreceptors and chomp into them. It's not quite that exciting, but they do grab bits and pieces of the shed discs and the shed material and recycle it. And it would be nice if they were really so simple and just straight lines. It kind of might remind you of the cells inside your intestine. These have the same function behind them. They want to increase the surface area of the cell so that there can be as many interactions with photoreceptors as possible. And our goal in this study was to provide a detailed description of a single cell out of the entire cluster of RPE cells, and describe their inner workings and their components. And because this kind of research is fairly sparse in this field, and I'll let you know a little bit about how we're doing this. A lot of the slides today were on micro-CT or X-ray data, but this data set we acquired through serial block face scanning, SEM scanning. And how that works briefly is that a sample of the retina, a little tiny cube, is embedded in a block of resin, and then SEM microscope takes one image, and then a diamond knife, shears off a very thin layer of the sample of the block. The SEM takes another image, and this gets repeated. And so you have one single section. This is what one of the single images looks like. And if you stack them all together in Dragonfly, you get a 3-dimensional volume. And this is not something that comes off the factory line. These are very individual samples, and they're biological samples, so there's a high propensity for artifacts or blurs or just things suddenly being different for biological reasons. And basically, what we wanted to do with this is to train a deep learning model to segment the organelles, and I'll tell you a little bit about how we did that. A lot of you know about the deep learning in Dragonfly. I'm just going to explain how we did it. We basically took the original EM data, the volume in Dragonfly, and annotated it. So ORS did this the first time for all volume, and it was a fairly 12 class, I think, model, UNet model. But basically, we used the annotated ground truth to train this model, and if necessary, add additional data to increase the power of the model until we got a satisfactory result. And that's one of the cubes that we got from ORS. So this is the entire volume segmented into the individual organelles. So in turquoise, the nucleus, the green stuff that you see, those are all these apical processes. It's not quite easy to see them now, but they're all individual, like hundreds of thousands of spaghetti intertwined. And so I'll tell you a little bit about how it might be counterintuitive to think about it in multiple classes, but I'm at least a proponent of using a lot of classes for your segmentation, a lot of labels, if you will. And because I've come to learn that the model does seem to perform better if you give it like a decoy or something to work with and to put things in, because if it's seeing things that it doesn't know, if it's like a foreground background segmentation, for example, then there's sometimes it doesn't get things quite right. But if you add a third class, a decoy or something else, then suddenly the model can offload all that like not super relevant data into that class. And that's something I did with this 11 class segmentation you see here. This is a few days work basically of training, and I was able to get quite a nice segmentation. And our models, or the ones that we use, are relatively simple because we have one computer, and all of these segmentations were done with an NVIDIA Quadro 4000, so not super powerful. So I've been quite jealous of some of the hardware I heard about today. So we were really pushing the computer to its limits with our five layers deep models. But yeah, I'll tell you a little bit about what we did to augment the data and everything later. But basically the motto we've heard before today is if you can see it in the EM or in the image, if there's a difference between contrast or something, you can segment it. And so for us, hardware was the limit, but it might be the case that eventually hardware, it doesn't matter. If you have a powerful computer or a powerful system, you'll still run into deep learning based problems or architecture problems. And so you could, instead of just segmenting tumor from healthy tissue, you might think about different sizes of tumors or different volumes of tumors to segment them that way, or all kinds of other different classes. And a few more tips that I have just from personal use. It might seem irrelevant or redundant to most of you, but it can help if you're just new to Dragonfly and deep learning especially, and you're just figuring out what to do. Like a lot of my research came from watching Mike's videos and just trial and error and just playing around with settings. So one thing that can be helpful is getting a mask for training and kind of focusing or showing the model where you want to have the attention focused. And so in example I'll show later, the nuclear pore complex is a very small point on the image, and it's quite underrepresented in terms of the volume on the slices. So having a mask that just covers just that small complex can really help improve the model. And in the similar vein, you could also add more ground truth, especially with these convolutional neural networks. That does seem to improve the the outcome. And in that vein, data augmentation also plays a role. I've done a lot of fidgeting around with that and experimenting what's useful, what doesn't really help that much, or it can just certain augmentations really slow down the epochs of the training, and it might be useful in the long term or later on. But just to quickly get started, if you just flip and rotate the data like 10 times, you'll get pretty good results right away for starting. And then finally, once you have a model and you feel like in the visual preview of your training, it looks good, I'd recommend maybe segmenting a small test chunk, a small volume, just so that you can see what it looks like in 3D, because that's what we were interested in, because that can show you weaknesses that the model has. It might be showing a lot of noise that you just don't perceive in the 2D view. So that's always a very dynamic process. You're training, and then you check out what the model looks like. You correct the results with more ground truth or different augmentation, and then we repeat the process until we get a nice segmentation. And so good ground truth does go a long way. So if you're a bit lazy sometimes, like I might be, and you do a bit sloppy segmentation, then those errors will propagate through the network and cause you some issues. So just take your time and do clean ground truth. Don't confuse the model. Now, I told you that we were interested in segmenting a single cell, and the way that we did this is, you'll probably shudder, we did a manual reconstruction by analyzing we tracked the origin of each of these processes of the RPE cell to the cell body and into their tips, including their branching points manually in TrackEM. This was before I had access to Dragonfly, and so a colleague of mine and myself, we spent about 200 person hours, or like 1.5 years working on this, and eventually we got a skeletonized 3D model of the apical processes and kind of the backbone of the cell, and then we filled in the cell bodies of those apical processes, which is in this second slide up here. And with Dragonfly, we were able to create a 3-dimensional mesh from that data, and then combining this with the segmentation from ORS, that they segmented the whole 3-dimensional volume. We cropped out the one cell to show the organelles and the contents of one cell, and that might be, the cell body itself might be fairly straightforward to separate out from the chunk, but these apical processes are all intertwined. And in the top left and the middle picture, you can kind of see that there are a lot of processes that aren't colored. They're just from neighboring cells, but they're all up in there. And so we manually separated all of them out and did measurements, and finally got our 3-dimensional version of the cell and could do measurements. And we calculated that there were over 800 apical processes that we manually traced, and they split up to, I think the largest we had was 15 or 16 times. They branch and continue into this subretinal space. And using this crop, I made a short video of the cell, just the EM volume, no segmentation at all. This is just what we made with Dragonfly to show the volume and show it'll cut through, and you can kind of see the top half is full of these big dark blobs, and the bottom is somewhat more diffuse, and that's this strong polarity that I was talking about, and we'll get to that in a second. And yeah, so these processes, they sprout up, so to speak, into the subretinal space, which is the space around the retina, around the photoreceptors. And you can kind of see in the image on the left that they have these gaps, and that's where the photoreceptors are stuck in, and there's an intimate connection between the two. And if anything goes wrong, then this waste from the photoreceptors keeps building up and leads to all kinds of pathologies. And so this is just a quick to show our segmentation, and then in the center, you have the actual organelles of the cell that we were able to quantify and do measurements with. Now, you might notice that there's these processes, they're not as nice and straight as the ones that we had before in these the schematics in the sketches, but that's due to something that's called compaction, which is an artifact from simply fixating the images, where the photoreceptors and the entire apical process will sink down and kind of fold together like if you're stepping on grass or one of these old toys where you press it and it claps together. That's sort of why these aren't nice and straight processes. So that's something we wanted to try to model and to maybe create a mesh with it, but that just, our computer said no, and it just wouldn't work on our current hardware. So we're still thinking about somehow straightening those out. But basically they're in this subretinal space that's filled with what's called the interphotoreceptor matrix. And I won't get into too much about that, but basically we trained deep learning models, all of these units, to analyze the distribution of the space, and we're able to segment a small test volume and figure out that actually from this space, almost half of it is this interphotoreceptor matrix, and this area is a specific target for gene therapy and eye injections. And if you inject too much liquid or too much material, medicine, whatever, it can disrupt the connection, this fine connection between the photoreceptors and the RPE cell. So that's something that needs to be thought about when delivering injections into the eye. Here's a little slide showing the rod and cone ratio. I used some models to segment this, and then manually chose the rods and cones because that would have taken too long to get done by the meeting. But this is basically just showing the ratio, and what we measured in our sample does follow the literature. It's about 2.7 to 1 ratio of rod and cones. And here's just another image from that crop, and you can really see the polarization of the cell. These orange brownish things are called pigment granules, and they generate a signal when you do OCT scans, which many of you probably have had just routinely done in eye clinics. This is what shows reflectance, and we wanted to analyze their volume and just visualize them because they're very important for this clinical imaging. And what we were able to do was to measure the volume and explain potential artifacts that occur in imaging, such as this halo that's kind of seen around the nucleus, and that might be explained by this ring of pigment granules, as they're called, that leave out the center. And when you look at it from the top, you can really see that there's a kind of a hyperfluorescent halo that we assume, or we theorize, is caused by these granules. Here's a different model I did. I was a bit of a modelholic, and kind of keep training new models to do new things. This one, I wanted to look at the internal structure of the mitochondria, which you can see on the bottom. You can see our model was able to quite accurately label the cristae and the little shells within the mitochondria. Unfortunately, we have quite high anisotropy, so getting it to display nicely in 3D in meshes is not always the best. This still an old slide, disregard that. Yeah, so I think this is a different present, never mind. So one thing that I was really interested in was going further down the cell was the nucleus, and you can kind of see it here slightly peeking out in turquoise, and we were able to really nicely see the nuclear pores in our EM volume, and as kind of a extracurricular activity on the weekend or sometime in my off time, I decided to just mess around with Dragonfly and experiment with some of these nuclear pores and see if I could get a model to correctly label them. And it worked quite well, and I took a volume and let it segment overnight, and when I came back in the morning, I was quite surprised to find that the nuclear pore complex was aligned in nice orderly rows, in parallel rows. And that's something that, as far as we can tell, hasn't been described before. It's been reported that these pores cluster in regions and they're like lumps, but nothing as distinct as this. And at first I wasn't sure if it was really doing the right thing. I thought it might have been some AI like superimposing its own, like, oh, this order has to be here. But then I did it on a second sample, on a foveal sample from a 52-year-old, which you see on the right, and that also had the same pattern. And using that same model, I was also able to segment all of the nuclei in the sample. And I think this was 20 frames tops for training, and within a few days, we could segment all of the nuclei in the given sample. But I don't know if you can see it that well, but you might notice something about some of the nuclei here. These two specifically don't have that order, and we aren't really sure why. It does seem to, so these pores, they do seem to follow the chromatin, which is the genetic material inside of your nucleus, and they do here too, but somehow it seems to be distributed differently. So we're still looking into that. We weren't really expecting to stumble upon this, and still trying to wrap our heads around why this is happening, if it only happens in RPE cells, or maybe only in humans and not in frogs or other model organisms. So we're still quite curious as to what's going on here. But I'll continue and go on to the next structure inside of the cell, which is the Golgi apparatus, which is often referred to as the post office of the cell, because it packages proteins into little vesicles and sends them to their correct destination. And you can kind of see it on the on this small slide. These little circles are basically where the vesicle passes through, and it gets a bit more information added to it to where it's going and packaged. And it's a structure that is around the nucleus in the perinuclear region. And our model was quite capable of segmenting it. And we made a short video showing just our cell with the Golgi apparatus in blue, in light blue, and the nucleus inside of the cell body, kind of cropped. I think these are all meshes, and this is all made in the Dragonfly Movie Maker. So again, pushing the limits of our hardware. There are a few little glitches like that. I don't think it's supposed to happen, but overall, I mean, we were quite impressed with the potential for this segmentation and for these architectures and deep learning. And continuing with a different sample, this is a Golgi apparatus from the 21-year-old fovea, which is a slightly different region in the retina. And it looks, unfortunately, the collection materials for this block face imaging weren't always the same. So this sample looks quite different. The organelles, some are better preserved, some are not quite as nice. Like in this image on the left, you can't really see the individual cristae in the mitochondria, but the Golgi apparatus is very well preserved, and it almost looks like a fingerprint once you look at it in the 2D slice. And three-dimensionally, I have a short little video where you can really see the individual stacks of cisterns where the protein passes through. And often in the textbooks, it's a very planar example where a protein comes, or a vesicle comes from one side and passes through and goes out. But this structure, if we look at it, it's almost twisted. So it's almost like it doesn't matter where the protein enters, it can always be calibrated to go to the right destination. I don't know, that's just my theory from segmenting this, but we weren't expecting it to work this well, but it did quite well. And and all these structures inside of the cell are connected by something that is known as the ER, or the endoplasmic reticulum. Won't bore you with the details, but we figured out that it does fill almost 20% of the cell body of the volume. So it's very important for having everything connected and transfer within the cell. Then I did a different model that was able to segment the actin filaments and the actin belt, which is the band around the bouquet of flowers. It kind of shows you really where all the apical processes sprout out, and it's like a nice little bouquet of flowers. And again, we were able to quite quickly train a model to do this segmentation. So I think almost all of these figures were done with different models, just because, like I said, I can't stop. And so we had the same model that segmented the apical processes was also able to segment these basal infoldings, which are like mini versions of the apical processes. And they're right above the circulation and the blood supply, and they're very important for, again, maintaining the health of the cell. And so even though the model at first, because I just didn't think of it, wasn't trained on the basal infoldings, it did quite well segmenting them. And then once we did train it, it was really able to show this like shag carpet view of the basal surface. And all of this is very exciting for us, because we were able to accomplish all this in a very short time with one computer and one or two people maybe working on it, on the deep learning part at least. And we were able to find out that combined with manual segmentation, these over 800 processes extend into the subretinal space and form these intimate connection with the photoreceptors. And basically our work has been more than cut in half by the deep learning aspect of Dragonfly. And so thank you from me to ORS. And here's a image of some of our collaborators from a meeting in Washington. And I wasn't there, but I'm here. So yeah, thank you a lot for your attention, and I hope it was interesting.
Well, that's very exciting, Max. I think it's very nice that we've had a lot of X-ray micro CT today, and we had a TEM talk this morning about cellular biology on the neuronal growth cones, and now we have an SEM talk about the RPE cell biology. And as scientific imaging biologists, we can appreciate that we can go asking questions and find answers to questions that we weren't asking, and discover things along the way. I mean, all disciplines can, but imaging biologists, they're just literally right in front of our eyes. And so you guys were treated today because the nuclear pore complex structure that hasn't been shown before, it's being presented at a meeting in 2 weeks for the ophthalmologist research, but the cell biologists haven't seen it yet. So what the cell biologists are going to tell us about how surprised or how unexpected it is to find these nuclear pore complexes that are organized, who knows what we'll learn and where it'll go. But it's very exciting. It's so exciting. It's serendipitous. It just popped out of you. You come in Monday morning and you look at the segmentation what is that? So that's super exciting. So thanks for sharing that. Now that I've talked for half an hour, questions from the group inside? Natalie, go ahead. So the question is about the sample prep for the cell tissue.
I can't speak too much to that because that was the University of Washington. Professor Dacey did that with his group. But as far as I know, they were just embedded in a resin that was then processed for serial block face scanning.
There's no high pressure freezing, at least from my notes with Dennis.
Yeah, I'm not sure what the material
but
was.
It's an osmium stain like everything else. So when I'm working with Max, because when we saw the data, people are often asking, how do we segment data? And I said, well, the tools will work if you have good, clean data. And so when they started showing me data, I'm like, you guys know how to produce good, clean data. How can I enable you to showcase Dragonfly with it? And so they keep producing good, clean data. So they keep feeding Max with lots of good data, lots of good images and results.
Yeah.
And it starts with sample prep, so for sure.
Yeah, and basically we or on our end, in our lab, we get the aligned and pre-processed kind of cleaned up images. So we basically can start straight away with making ground truth and training the models. So I'm coming out of, I kind of follow the slippery slope into research, and so I'm not that well versed with the cutting, but yeah.
Do we have other questions from inside the room? All right. Well, Jess, any questions? All right. Well, thanks. So that was some really exciting cell biology, and I can't wait to see what the cell biologist and the cytologist have to say when you guys present that shortly. Thank you so much for your time, Max. That was fantastic.